In this video, we will discuss about the inducible AMT, whether it can suppress the cancer progression or not. The advantage of inducible AMT is that the researchers will have better control when to induce this AMT to suppress the cancer progressions. So let us first understand their design, how they design their inducible AMT system. So they designed this RTTA guide RNAs using AAV, where they put this RTTA, it is tetracycline reverse transcriptase here, and they have this TRE, which is also placed just before this uh, DCAS9 VP64 promoter, and this is called as TRE DCAS9 VP64 AAV. So what will happen when these two AAVs are transfected into the cells and the doxycycline is introduced. This because of the doxycycline introductions, what will happen, this will get translated and it will interact with this doxycycline, or we can say this tetracyclines, and it will then cause the translations of this TRE DCAS9 VP64 AAV. So when the doxycycline is not there, this RTTA is translated, but due to the absence of doxycycline or tetracycline, it does not forms a complex with it, and it will not come and interact with this TRE to help in the translation of this second virus. So when all these three things are present, that is the RTTA, guide RNase AAV virus, and TRE DCAS9 VP64 AAV, and also these doxycyclines, then we can see this DCAS9 VP64 signals. And when they transduce this inducible system, into the SCID mice after this tumor injections for 4 weeks, and they check the tumor size and other proliferations, glycolytic genes and fatty acid oxidations 3 weeks after the injections of this two viruses or adipose with this inducible AMT. We can say that inducible adipose. Then what we have seen is that in the case of control, where the tumor says where the tumors are co-transplant with this control AMT, they see some tumor size, but when the tumors are co-transplant with this UCP1 genes upregulated AMT, they are able to see decrease in the tumor size. So in this case, it's an inducible AMT, that is co-transplant along with tumors. Next, they check the proliferation markers, that is MKI67. It is also goes down when both the adipose with UCP1 genes upregulated is co-transplant with the tumors, compared to the control case where VP64 genes is co-transplant. And the metabolic genes such as the glycolytic genes, GLUT4 GCK also goes down, and fatty acid oxidation genes, that is CD36 and CPT1B, also goes down in the experimental case where AMT has these UCP1 genes upregulations are co-transplant with the tumor cells. Next, now they prepared different methods to generate this organoid of this UCP1 genes, CRISPR adipose organoids. Here they use this polycaprolactone microbial scaffold, and in this case the transplantation efficiencies of this organoid is higher. So when they use this approach to transplant the adipose organoid into the tumors, in the adjacent of the tumors into the mice, what they are able to see that, so first this we will understand their experimental plans. That is, first the cancer cells are infected into the mice using xenograft method. Then after 4 weeks, this organoid based on their new microbial scaffold is implanted adjacent to the tissues, and after 3 weeks they check these following parameters. In the panel E, we can see that the cellular scaffolds that are shown in their map, and those are scaffolds are this co-transplant co-transplant or we can say transplant into the mice, and this is the how the tumors and cellular cell scaffolds looks like adjacent to each other. When they check the tumor size, they are able to see that tumor size goes down in the when the tumors is co-transplant with this adipose with UCP1 genes upregulations, and these things are quantified on the right hand side of the panel F, where we can see that tumor size goes down. Then they check the other parameters such as proliferation markers, it also goes down, that is MKI67, and the metabolic genes, that is glycolytic genes, GLUT4 and GCK also goes down, and the fatty acid oxidations related genes, that is CD36 and CPT1 also goes down. So this indicates that both of their approach, that is inducibles, AMT, and also their advanced microbial scaffold for organoid formations has the positive impact or has the consistent results that they have seen in the previous method. So this gives a more better method for the researchers to control when to induce this AMT and when to suppress the tumor progressions. So I hope you understood their inducible AMT system and their effect of their new microbial scaffold system for these organoid formations of the adipose with these UCP1 genes or for control BP64 genes upregulations. If you want to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.